Good morning, ladies and gentlemen.
My name is Indra Faizatun and my partner
Novilla Dwi Candra. We are students of State Islamic Senior High School 1 Kudus, Central Java, Indonesia.
We are going to present our research entitled BABL-Caf, the enhalocrase from Seagrass Seeds Enhalus acoroides, Latoh Caulerpa sp, and Mocaf flower, rich in antioxidants as today's healthy staple food source. So, let's get down to our business. Ladies and gentlemen, the question of our experimental research is, which composition affect nutritional value on enhalocrase BABL-Caf? The variables of this study are as follows. The independent variable is the material on enhalocrase, Seagrass Seeds flower, Mocaf flower, and Latoh powder, while the dependent variable is nutritional value. Based on the question above, the hypothesis that we made is that Formula 1 rich in antioxidant, low in glucose, glycemic index, fiber content, and good proximity analysis.
Rice demand in Indonesia increases each year in line with the increasing number of people who consume the majority of rice as a staple food. The decline in rice production takes new innovations to avoid problems that occur by means of making enhalocrase. The main raw material in the manufacture of enhalocrase is Seagrass seeds. Latoh is also rich in compounds that are beneficial for health. Besides the Seagrass seeds and Latoh, complementary ingredients in the manufacture of enhalocrase is Mocaf flower. Seagrass seeds, Latoh, and Mocaf flower are very supportive to be used as ingredients in the manufacture of enhalocrase because contain rich in antioxidants. Antioxidants help to provide health protection to the body, which is the ward of free radicals. The manufacture of enhalocrase from Seagrass seeds, Latoh, and Mocaf flower is one of the innovations to limit Indonesia's problems that often import rice and replace Ciherang varieties because of failed crops.
The equipment of these studies consisted of extruder, grinding machine, baker glass, ATM sheaf, spatula, porcelain cup, and others. The main material for this research include Seagrass seeds flower obtained from Kepulauan Seribu, Jakarta, Mocaf flower obtained from Bandung, and Latoh Caulerpa sp obtained from Jepara.
The procedures of this research are as follows. Seagrass flower making process. First, sorting to choose seagrass seed with good quality. Second, seagrass washing and size reduction. Then, drying to obtain the optimal form of flour at temperature 55 degrees Celsius. Next, smoothing using a grinding machine to make flour. And the last is sifting using ATM sieve to get final Seagrass seed flour.
The next is Latoh powder making process. The first, cleaning and washing. The second, immersion. The third, drying until the water content reaches 20%. The next, size reduction. And the last, the sieve uses ATM.
Next, enhalocrase making process. First, formulation aims to mixture of enhalocrase raw materials made from Seagrass seeds, Latoh, and Mocaf flower with the desired composition. Second, preconditions. The mixture of raw materials result from the formulation was maintained in warm conditions, temperature 80 to 90 degrees Celsius, then flowed to the extruder. Then, extrusion. The dough will be reheated at temperature 120 degrees Celsius. It was done with double screw extruder, which had the ability and greater flexibility to control the process parameters and products. Next, drying. Drying is done by using dryers. And the last is packaging. The enhalocrase product packaged in tape and vacuum plastic to prevent the mold and bacterial growth.
The next is analysis. Nutritional value that we analyzed in this study include antioxidant, approximate, glucose, glycemic index, and fiber content. Antioxidant analysis. Antioxidant activity was carried out by using with DABL-pH method in spectrophotometry by reacting several types of samples with a DABL-pH solution at a wavelength 517 nanometers. The result of antioxidant activity was computed as follows. The result of several types of enhalocrase showed that antioxidant levels in the Formula 0 treatment was 17.71%, Formula 1 was 80.52%, Formula 2 was 52.50%, and Formula 3 was 74.27%. The differences in antioxidant test results were influenced by ingredients from Seagrass Seeds that cause high levels of antioxidant. The higher antioxidant value, the higher the shelf life of food product and can protect food product from deterioration of quality processes caused by oxidation such as rancidity. Antioxidant could resist free radicals found in the body which were obtained by the body's metabolism, air pollution, food pollution, and sunlight. Antioxidants were also needed to prevent oxidative stress in the body. Oxidative stress was a condition where an imbalance exists between the amount of free radicals and the amount of antioxidant in the body that can be harmful to the body. So, the body must consume food with high levels of antioxidant.
The next is proximate analysis. First, moisture content. Moisture content is a lot of water content in the ingredient and expressed in percent. Low water content in an ingredient causes the ingredient to have the most robust resistance from the threat of microbial attacks and decay in the near term. It can be concluded that in the Formula 0, Formula 1, Formula 2, and Formula 3 have a lower water content than rice in general, that is 11.22%. So, food security in Formula 0, Formula 1, Formula 2, and Formula 3 are stronger than rice in general.
The next is ash content. Measurement of ash content aims to provide an overview of the content of internal and external minerals derived from the initial process to form extract. The high value of ash content in the Formula 2 and Formula 3 is caused by the high proportion between Seagrass Seeds and Mocaf flour. It can be concluded that the ash content in the Formula 0 and Formula 1 is good for healthy food for analog rice that corresponds to the ash content in the rice generally.
The next is fat content. Fat is an important dietary substance that contains energy sources more effectively than carbohydrates and proteins. According to BABL-Caf 2011, the fat content in food products is categorized as low if its value does not exceed 3%. Fat is required by the body, but excessive consumption can cause obesity that can trigger the onset of various types of diseases, such as diabetes and atherosclerosis. It can be concluded that in Formula 0, Formula 1, Formula 2, and Formula 3 have a lower fat content than rice in general, that is 1.37%. So, it can be used as a reference for healthy food in analog rice.
The next is protein. Protein is a food substance that is very important to the body because it can function as fuel, builders, and regulators in the body. Protein is the largest component after water as well as the source of amino acids containing elements of carbon, hydrogen, oxygen, and nitrogen that are not owned by fats and carbohydrates. Proteins are important in the formation of new tissues, enzymes, hormones, antibodies, and other functions in the body. It can be concluded that in Formula 2 has higher protein levels than rice in general, that is 8.66%, so that it has healthy food quality in analog rice.
And the last is carbohydrates. Carbohydrates are a common chemical content found in foodstuffs and are the main source of calories. Carbohydrates are useful to prevent the onset of ketosis, the breakdown of excessive body proteins, loss of minerals, and are useful for forming fat and protein metabolism. It can be concluded that in Formula 2 have a lower carbohydrates level than rice in general, that is 78.19%, and lower carbohydrates will prevent obesity.
The next is glucose. The result of the first analysis of glucose levels without using acidic hydrolysis method obtained a glucose levels standard of 1000 ppm. Glucose levels in Formula 0, Formula 1, and Formula 2 are not detected in the standard range. In Formula 3, glucose levels are detected at 71 ppm. The result of analysis using the acid hydrolysis method obtained the standard data of glucose levels in 1000 ppm. Glucose levels in the Formula 0 treatment was 0 ppm. Formula 1 was 273 ppm. Formula 2 was 687 ppm. And Formula 3 was 644 ppm. In the Formula 1, 2, and 3 have a glucose levels under the standard data of glucose levels of 1000 ppm. This is because the sample has not undergone enzymatic glucose breakdown, so that glucose cannot be identified perfectly on the HPLC tool. This data showed that it was obtained glucose levels close to the standard. This is due to the acid administration formula of sulfuric acid in sample. The result of this study also showed increased reaction time resulting more and more glucose. This condition occurs in all temperature treatment.
And the last is fiber analysis. The lowest number of crude fiber analysis is in Formula 1, which is 4.56%. The lower the crude fiber, the higher the food fiber, because it can slow down the digestive velocity as well as slowing the emergence of blood glucose. While the highest crude fiber content in Formula 3 is 11.2%. The highest food fiber can prevent from various diseases.
The next is conclusion and suggestion. In conclusion, we get that the proper treatment recommendation in Formula 1 because the data on the result of the analysis of Formula 1 average better than the other treatment. And to future research, the suggestion that we can give is combining the material of making analog rice that has more precise characteristics so that the analog rice produce better quality and functional.
That's all our presentation.
Thanks for watching.